All right, let's go ahead and get started. Thanks, Wall, for that awesome intro, and thank you to Amy and Joanne for organizing this awesome workshop. I think I'm really excited to be here and really excited to start talking about processing in cryo-EM with you all. This is obviously not a cryo-EM microscope, but use your imagination. So the next 2 days are sort of belong to Structura, which is where I work, along with Michael McLean and Ollie. They did fly across the country to be here. I am based in Portland, so I kind of flew halfway across the short way. So you don't have to be as thankful to me. I wanted to highlight that my role is the application scientist for education and training. So this really is my favorite part of the entire job, the entire kind of world of science, is using what I know to help people get the best results that they can out of their data. So I really do encourage you to ask me questions while we're here for this workshop, as well as, Structura makes available our discussion forum where I am basically 24/7. And I brought my little yellow notebook here so I can write down anything you ask and hopefully improve our educational resources so that it's easier for future people to learn. So the things that we're going to do, we being the Structura team, are we're going to review some foundational concepts and theory of cryo-EM in a lot of people's kind of like applications to this workshop. There were a lot of requests for discussion of theory. I think we have a bit of a mix in terms of experience, but it's good to make sure we're all on the same page before we move on. My other goals are to help develop some intuition regarding map pathologies. I think that this is a skill that you develop just by doing cryo-EM, the ability to look at a 3D map and see, at first you just look and you're like, this is bad. And then you kind of learn, okay, it's bad in this way. And then you kind of learn when it's bad in that way, it usually means this. I'm going to try and help jumpstart that process for you so you can develop that intuition a little faster. And then finally, basically every single person, flexibility and other types of heterogeneity. I think that is one of the huge advantages of cryo-EM over some of the other ways of getting 3D structures of macromolecules. So we'll definitely be talking about that some, and I bet some other speakers will too. If you, like me, are the kind of person who falls asleep during lectures and workshops, just pay attention now, and then you can go right to bed. I'm just going to kind of give a few of the really important points that I think are really something that I really want you to take away from this. The first one is there really isn't a best way to process data. There certainly are some wrong ways. I'm going to try and guide you away from those. But at the end of the day, there's always going to be 3 or 4 different ways that you could get to certainly a usable map. And so I want you to really take to heart that you're kind of trying to get to a map. And in the path you take there's probably a couple ways to do that. As part of that, I think you should try everything and see what works. I think can be really kind of fun and exploratory. And so I recommend that anytime you think, oh, I wonder if this would work, try it and see. It might and it might not. That's okay. And it's okay because we learn by making mistakes, right? I remember when I was first starting to learn image processing in cryo-EM, I was terrified of running the wrong job. I didn't know what was going on. What the hell is motion correction? And I think when you run one of these jobs and it doesn't turn out how you expect, that's okay, right? You learn something, just like you would learn something if an experiment at the bench failed. One of the final thoughts I want to leave you with is to really think about the images, right? When we collect cryo-EM data, we collect these horrible movies, and then we motion correct them into these horrible micrographs, and then we pick and extract these horrible images of particles, and then you get a beautiful 3D map out of them. So it's really tempting, because most of what you look at is the maps, and because the maps look so much better than the disgusting images, it's really tempting to only think about the volumes. Unfortunately, I think it's best to think about the images, right? Because what we're actually doing is performing these image alignments that we'll talk about in a bit. We're classifying the images, right? And so I think if you really focus when you're launching a job on what's happening to the images, you'll have an easier time kind of figuring out what the next best step is. And so throughout, I have some prompts to think about kind of what these jobs are doing to the images themselves. This is a part that I think a lot of people are not going to like, called think, pair, share. And I'm bringing it up now because it's something I'm going to force you to do. So think, pair, share is an educational technique in which I pose a question to the learners, that's you guys. The students think on their own about the answer to that question, what they think the answer would be. Then you're going to pair off. You could do 3 if you're near 3 people to discuss your answers with each other. And then typically you would share with the whole class. Lucky for you, too many people here to share. So we're just going to do think pair. After we do the pair, so after you discuss with each other, I will kind of give what my answer would be. But again, there's no right way to process data. So my answer is not the right answer. It's the answer that I kind of, through experience and through research, have come to, but I'm sure there's others. I'm worried that this microphone is not. When I ask these questions, they'll kind of be formatted like this. We'll get a question, and then there'll be, there'll be a timer that'll hopefully have a time on it, and then there'll be another one there. I'm sure the other ones will work. I know this feels awkward, and you're all probably pretty mad. I wouldn't force you to do this if it didn't help, right? And because we're all scientists, here's research, okay? The experimental group that did think, pair, share did better on their exams than the group that didn't, okay? It really does help, and I'm sorry. I'm just following the research, okay? The last thing I want to say is that students who explain their thinking benefit the most, right? So thinking on your own is helpful and discussing is helpful, but the most helpful thing is explaining why you got to that answer. And the reason I bring this up is I really want to encourage you when you're talking in the pair to make sure that everyone gets a chance to explain their rationale, right? I really think it's good to be conscious to make sure that you're not just explaining what you think and expecting the other person to listen. I really want to encourage you to have a conversation. All right. I know there are a lot of remote people watching, which doesn't make me nervous at all. And so these exercises might be challenging, right? If you're remote and you're watching on your own, but I think they're still valuable. There's a few ways I think you can more valuable if you're watching. Is the microphone cutting in or no? Or am I? Well, one is if anyone else in your lab or institution is participating remotely, you could maybe watch the workshop together and just participate in them sort of as intended. If you're participating remotely and you're kind of alone, you can, during the share portion, just write out your thinking on the piece of paper. Like I said, the explanation is the most important. And I think kind of that process will still sort of occur if you write out the paper. I don't have research for that, though. Okay, just a little bit more of kind of meta discussion, and then we'll get into it. We have kind of formatted our section in terms of case studies. During our part of this workshop, we're going to present 4 case studies. Each one is, we're going to start with a data set that's publicly available on Empire. And we're going to kind of work through it step by step, job by job, telling you what jobs we run, and importantly, telling you kind of why we ran those jobs, right? What about how the data or how the map or how the particles are looking caused us to run this job? Why did we pick these? Again, I want to emphasize, I'm going to be showing you a lot of jobs and a lot of parameters. A lot of these things are sort of, when I created the job, this is what worked. There are parameters that are pretty sensitive, and I'll try to highlight those where it's like, this has to be kind of set to this exact value. Some of them, I'm showing you exactly the job I ran. So it might be, oh, actually, if you change that parameter a little bit, it would still work, right? Like I said, there's no best way. You just got to find the way that works. Just like an experiment at the bench, every job teaches you something, right? So you launch some job, it gives you a map that looks horrible. You can sit and think, why did it look bad? What did that job do to the particle images, right? And then why did that make the map look worse? And so what job should I run instead? Okay. So now, what are we doing today? We're starting with the fundamentals of cryo-EM image processing, just to make sure we're all on the same page, as I said. Then we'll move on to the first case study, or really pair of case studies on some ion channels. And those we'll kind of use to talk the basic processing pipeline for cryo-EM images, as well as some basics of symmetry. And then finally, Michael will take over and we'll talk about encapsulated ferritin, which is a really cool data set. And I think Michael's done a great job putting it together, where we're going to talk about some exciting things, including non-point group symmetry, which is challenging, but really cool, I think. The last thing is, please hold your questions to the end, or at least I'll have a slide that looks like this when I need questions or want questions. Normally, I'd encourage you to interrupt me, but there's a lot of folks here, and for the people participating remote, want to make sure that everyone has a chance to ask questions. So please hold your questions to like designated times, and you can write them down just if you need the context. Okay, so with all that out of the way, we can start to talk about Cryo-EM fundamentals. Make sure I know how to read my watch. Okay, so first, let's talk about data collection and pre-processing, right? This is kind of the very first step of any Cryo-EM image processing pipeline, is getting the images that you're going to process. And unfortunately, there's a lot here that's wrong, but some of it we can correct with software. So hopefully you've all collected a data set before, but if not, you have a grid. They are 3 millimeters-ish across. And on the grid, there's these metal balls. Those metal scopes that have holes in them, right? Those holes have your particle in them. Easy. The particle is just suspended in this amorphous ice, and you hope that it's in a random orientation. And the reason you want it to be in a random orientation is so that when you look at all of your particles, you've essentially looked at the sample from every point of view, right? If they're kind of evenly distributed in all the different poses, you've kind of seen all the poses by the time you've seen all your particles. Ideally, your images kind of look like this, where you have a pretty even spread across the micrograph of where your particles are, and they're all in random orientations. If they're too sparse, have to collect way, way more images to get enough to make a map. And it'll be harder because you have more space that doesn't have your particle. You'll have more junk picks as well. If they're too crowded, the particles will overlap. And that can cause problems with picking and alignment, things like that. The last kind of problem that is probably the most onerous is orientation bias, where all of these particles are in the exact same orientation. Like I said, you need that random orientation to make sure you've seen all this kind of sides of your volume. And so if you have orientation bias, that problem is pretty hard to solve because we just don't know what the other poses look like. There are ways, So kind of stuff you might have to go back and collect new samples to resolve. There's some other things that happen once you actually expose your sample to the electron beam. The first is motion. So before you expose your particles to the electron beam, they're kind of nicely clustered in your little square. Unfortunately, the beam causes the ice to bow, the stage to shift, right? So you have this movement and spread of the particles once that electron beam hits them. So that's why we collect in movies, and then we perform motion correction, where we take each frame of that movie and kind of try to line it up, put the particles back in the same position before we average them together to a micrograph. The other thing the beam does is it irradiates the sample, perhaps obviously. But what happens when you irradiate the sample is you're just bombarding it with these high energy electrons, which quite literally burns away some of your sample, especially the high resolution parts. Okay, so this is basically the high resolution information especially is very dose dependent. The early frames of your movies will have higher quality, high resolution information than the later frames. And so because of that, during motion correction, we also do something called dose weighting, which is where we say, trust the high resolution information in those early frames more than you trust the high resolution information in the later frames. And again, this generally happens automatically, but it happens. One of the things we do to prevent the, or to sort of lessen the effect of that irradiation burning away that high resolution information is we collect with low dose, okay, right? Fewer electrons mean that high resolution information lasts longer than if we were using a ton of electrons, okay? The problem is low dose makes our images look really bad, right? This would be a really, really high dose. If the protein could survive that much electron dose, you'd be able to tell really easily what orientation it's in. But we actually collect images that look like this or worse, right? Because we need that really low dose so that our protein lasts as long as it can. But because of that, the signal to noise ratio is really poor, right? And so it makes it harder to figure out what the pose of the particle is, what rotation and translation has been applied to that particle. The last kind of thing that happens when you collect those images is that we need to generate content, okay? One of the big problems with cryo-EM is that protein and water, from an electron beam's point of view, are basically the same thing, right? We don't have this, they absorb or scatter electrons essentially the same amount, okay? So there's very little contrast in a cryo-EM image by default. To induce contrast, we collect at defocus. And this is a kind of confusing and very physics-heavy topic, which we don't really have time to get into here. Ollie can testify, I spent about a month driving myself crazy learning about the CTF, so I'll spare you that. Grant Jensen has some really nice videos on YouTube if you're interested in the topic to kind of get some background. But the basic principle is we collected defocus to get some contrast. And if you've ever used a light microscope and looked at something like a cell on a slide, if you're exactly at focus, there's not very much contrast, right? But if you move a little bit away from focus, either up or down, these dark and light rings move away from the cells. We're doing basically the same thing in Cryo-EM. This is a simulation where on the top half, I have not applied any CTF to this so-called sample. It's just a cross grading, basically. On the bottom half, I have applied the contrast transfer function, the CTF. That's the function that tells us what our final image will look like after it's been through the beam and the lens and everything. I'm going to play an animation where I increase the defocus, where I move further away from the focal plane. I want you to pay attention to two things when the animation starts. One is in the center of the image where we have the low frequency parts of this grading. I want you to pay attention to what the contrast looks like there. But even more importantly, on the edge here, where we have the high frequency stuff and then no particle anymore, I want you to pay a lot of attention to what happens down here, right? We can see with no defocus, we can actually see the high resolution stuff a little bit. I'm gonna start the animation now. So there's a few things to pay attention to, right? One is in the middle, suddenly that low contrast information has jumped out at us. That's what the contrast transfer function does. It turns defocus into good contrast at low resolution. The other thing that's happening though, is on the edges of the particle, right, where that high resolution information and then the edge of the particle is, suddenly that signal is going way outside the actual bounds of the particle, right? This particle is not changing at all, but the defocus moves that signal, moves that high frequency signal outside the physical bounds of the particle, okay? This is a really important concept because if you just extracted a particle image that only contained exactly the real physical size of the particle, all that high resolution information that got pushed out of the image would be lost. And so that's why we extract with a bigger box. We'll talk about extraction in a second. The last thing to notice is that if you pay attention, there's in the bottom a few vertical lines where it stays gray. That's where there's zero contrast. The reason for that is the CTF oscillates up and down, positive and negative. Anytime it crosses zero, you have no contrast at that frequency. And you can see as the defocus changes, those vertical bars of gray where the contrast is zero move back and forth. That's why we collect with a defocus range when you're collecting movies. Because if you collected only one defocus, those zeros would always be in the same place. So you'd always have zero contrast at the same frequency. Because if you collected a range, that is spread out. And so across all your particles, you have contrast everywhere. So that is the kind of quick and dirty summary of data collection and pre-processing. First, we freeze the samples in ice that's suspended in a cryo-EM grid. And you want those to be kind of as closely packed as they can be without overlapping or sort of touching each other. And you definitely want the orientations to be distributed kind of randomly. Okay? The electron beam degrades the image quality, right? Those electrons are just burning away that high resolution stuff. So we need to do this motion correction to correct for what the electron beam does to the ice. And we also need to collect with low dose to preserve that high frequency information. The last thing is that contrast is very poor if we collect at focus. And so we collect with defocus, which we have to computationally correct by modeling the contrast transfer function and sort of applying it in reverse. And then we also need to extract that bigger box to collect that information that's been pushed outside the particle bounds. Okay. Maybe, no, we can keep going. Sure. Yeah, okay. Let's do some questions really quick about pre-processing. That's all the stuff we've talked about so far. Freezing the grids, motion correction, contrast transfer function. Or any. Let's wait for the microphone just so people online can hear the question. Thank you, then.
Thank you. Yeah, so I have a question about when you talk about the defocus and does it make a difference if you just take one focus value and talk with that, or maybe do a range of defocus values? But then if you do a range, that obviously is how do you then put it back together?
That's a good question. Yeah. So the question was basically, if you do a range of defocus, I recommend just one focus. Doesn't it? So I think there's kind of two sides to that. One, I think you don't really have another choice. The reason is those zeros. So if you always collected at the same defocus, you would just have absolutely no information about certain frequencies where the contrast transfer function is zero. So even though ideally you'd be able to kind of just either collect at focus or pick one defocus and stay there, I think in practice, you unfortunately can't really do that. In terms of doesn't that make it more complicated? I think yes and no. I think at the end of the day, talking about the black box where you push a button and get your candy, the CTF, the contrast transfer function estimation, I think is the most push a button and get your candy job there is. It generally just kind of works because even though it's a complicated physics topic, I think it's relatively simple to model in the computer. And so you can run the job and no matter what the defocus is for all your micrographs, you'll kind of get the right answer. So I think you're right that it is a little more complicated, but not to the user, I guess. Yeah.
I can actually just add to that just a little bit. So the other point about the CTF, Rich already mentioned, the zeros are one problem that you can't collect everything at the same defocus. There's also a physical reason for that. When you're actually imaging the sample in the microscope, you can't focus the microscope exactly precisely the same every time, because the sample itself also has variations. The defocus is related to the lens power and so on, but it's also related to the physical position of the sample within the column. And so if the sample's a few nanometers up or down, that's a change in defocus that shows up in the images. So effectively, no matter what you do, you still have to estimate and correct the CTF separately for every single micrograph you collect, and ideally even for every single particle that you collect. And so that has to happen no matter what. So you may as well collect your data at a range of defocus values to avoid the zero crossing problem. The other thing is computationally, correcting the CTF is relatively straightforward if you can estimate it well, as long as you get a good estimate for every image. And this comes back to another point that Rich mentioned about crowding and the sort of density of particles within a micrograph. The way the CTF is estimated is by looking at the actual image that you got from the microscope, an entire micrograph, and estimating those rings and everything that you saw in that image in the video that Rich showed. Those rings, we can actually see them and we can measure the spacing between them. And that's how the CTF is actually estimated. And so if you have more particles in a single micrograph, you will see more of those rings. There's more information about estimating the CTF. So it's easier to estimate the CTF when you have many particles in an image. Even if an image is very crowded, you can actually estimate the CTF very well. The crowding will have other problems later on, but it makes it easier to get defocus estimates, which is something you have to do no matter what, because you can't focus the microscope precisely. So that's just another thing to keep in mind.
Definitely.
We do have a question from the Zoom people.
Yeah.
And two questions, and I think they can be combined into one the way it's written.
Great.
First is, how big should the box be around the particle to get that contrast? And does this change the amount you increase it? Does it change with larger particles versus smaller particles? Is twice as big good one way, but 3 times as big is better the other way? And the other question is, why does the high resolution stuff move out?
Oh, okay. See, this person is trying to get me to spend an hour talking about the CTF. So I'll tackle the first one first. How big does the box have to be? We'll talk a little bit about that in later slides when we're talking about particle extraction. But the general rule of thumb is 1.5 to 2.0 times your largest particle width, like your biggest diameter. That's, I think, pretty reliable. I would say I generally do twice as big, just because I don't care about our hard drives. But there's an equation you can pull up. Rosenthal and Henderson, 2003, I think is the reference. But I'm sure if you just Google how big should my box be, you'll find it. Where you can calculate based on the accelerating energy of the microscope and the size of the particle. The other thing is it depends on the defocus, which is why I brought this back up. Right? You can see that at lower defocus, like here, where we're at 1.5, this information is spread very wide. But as we're closer and closer to focus, the shift is less and less, right? So it depends on your average defocus. If you've collected a data set with very high defocus, maybe you went all the way up to 3 microns or more, right? You're going to need to extract a bigger box than if you're very close. Yeah. So, and then the second question, why does it happen? Great question. If you figure it out, let me know. I think that one is going to be hard to answer succinctly. I think a good place to start thinking about it is it's so complicated. I think I would read some papers about it. It's very complicated.
Delocalization.
Yeah, just Google signal delocalization, and you'll be stuck in the same morass that I am.
Great questions. Thanks.
Yeah, thank you. I think we have one in person.
A very nice question.
Uh-huh.
So how do you decide what is the best defocus range for your data collection?
That is a really good question, which I'm not sure there is an answer to, and I certainly don't have one. I think there are sort of rules of thumb where you kind of stick around the, now I'm going to say what my rule of thumb was when I was collecting data, and then immediately everyone's going to disagree with me. But I think generally in the like -1 to -2 micron range is sort of typical, maybe a little more than that, maybe a little less. But it depends a lot on your sample. I think typically smaller samples, you're going to want to collect with more defocus, because I think one of the big things that defocus does, if you look at our little animation here, is it makes the low resolution signal more intense, right? It increases the contrast at low frequencies. Low frequencies are most useful for particle picking. So if you have a small particle with poor low resolution signal, you're not going to be able to see it, right? If it's big and you have kind of bad low resolution signal, it's okay because it's huge, right? It's a ribosome. You'll find it. So I think generally people will collect with more defocus the smaller their sample is, or maybe the noisier their data, or if you have very thick ice, you might want to collect with more defocus. But that's something that you'd probably want to talk to the wonderful people at S2C2 and have them help you figure out the best answer for your sample.
One more question. So also, like, if you're collecting the data set for the first time, let us say, so how do you decide like what is the best electron dose like for your data collection?
Yeah, that's another good question. What's the best electron dose? And again, probably something that's a little bit sample dependent and a little bit dependent on what exactly you're doing. I'm ashamed to say I'm not super up to date on what people are doing these days. When I was collecting data, we would collect with like 50 to 65 electrons per square angstrom. I think that is one that I wouldn't follow my recommendation. I'd see what people told you who use the microscopes more often than I do. Yeah, but again, it's going to be sample dependent. It's going to be data set dependent, unfortunately. I'm not sure how exactly when you would change it or when you wouldn't, unfortunately.
Okay, maybe I can help.
Yes,
you answer
definitely.
That. Okay, so as a rule of thumb, if you want to get beyond 3 angstroms, I would strongly recommend your total cumulative dose no more than 20 electron per square angstrom. And the next question you ask me, how do I know? Well, because I'm old enough to know. That's one answer. The other is, in the old days, we have done a lot of radiation damage study of biological samples using a protein crystals. So a lot of these so-called critical dose is based on those studies. And so you can actually do an experiment yourself by dosing it differently. And the one good thing about using the direct electron detectors is you have multiple frames. And then you can use all the frames, which even though the later frames have more damage, but then you still have better statistics. So that means the image is highest signal to noise at low frequency, because the low frequency information is very useful for determining the particle orientations. But when you do the reconstructions, you may not use all the frames, but you may only use some earlier frames, which represent a less damage. Now, I think there's some recent studies in the last 10 years or so that in the very beginning of the frames, so 1 or 2 frames, they're actually turbo image, because the thing moving so badly, you cannot correct it at all. So that means some of the virgin particle with really minimum to no damage probably don't exist in some cases.
Mhm.
Okay, so if you want to be more references, you can come to me and I can show you that in fact we have a paper just describing more recently how some of the damage are affected.
Yeah, and I think that's a great point about them being movies too, is that as long as you're not, each frame doesn't have very many electrons in it, you are kind of in control of the final dose, especially with EER is another great sort of advance in that sort of realm. The other thing too is there are jobs, so CryoSPARC calls it reference-based motion correction, Reliant calls it particle polishing, but the idea is similar, where you're basically calculating. So I told you about dose weighting, where we sort of say, trust the early frames, high resolution more than the late frames. Both of those jobs do empirical dose weighting, which is basically, we say for this data, for this movie, for this exact sort of particle, well, I guess for these movies really, how we fine tune that, right? How much more should we trust the early frames? And sometimes, as Wad just said, the first frame is actually bad, actually very bad, because it's moving too much. And so our sort of intuition about the first frame should be the best and the last frame should be the worst is sometimes a little bit off. And so there are jobs that can help you in that area too. Yeah. I think maybe let's do one more and then, unfortunately, but there will be questions at the end too. So if we don't get to you. Yeah.
I wanted to go back to the defocus again.
Yeah.
Say you have a protein, one rigid part and one floppy bit. The rigid part is easy to get, but for the floppy bit, like how should I change my defocus to actually be able to see it?
Yeah, that's a good question. So generally, the contrast transfer function doesn't change too much on the scale of a single particle. There are exceptions to that. Big viruses are the ones that come to mind, where they're so big that you're actually at kind of meaningfully different defocus across the thing. That's called Ewald sphere correction, if you're interested. So for things where you have like a flexible domain that's hard to resolve, I wouldn't worry too much about the defocus. I would kind of collect a sort of normal, and what that means will depend on the sample, right? But like kind of a normal defocus set of movies. And then there are other techniques you can use to help resolve that flexible bit, things like local refinement, particle subtraction, flexible refinement, 3D variability. And we'll talk about all those during the next 2 days. So I think there are more like kind of computational based approaches that I would take, as opposed to a kind of physical change, something about the microscope approach. Yeah. Great. Okay, thanks for all those awesome questions, everyone. And as I said, there will definitely be time for more questions at the end. So if you had questions about pre-processing, we didn't get to them, I really encourage you to ask them at the end. So now we have our movies that we motion corrected into micrographs, and our micrographs have a contrast transfer function estimate. So we say, for this movie, we think we were at this defocus, which means we need to multiply by these values, et cetera, et cetera. What's the next step? Well, the next step is we need to get our particles out, right? Here's our micrograph. I can point and say, look at all those blobs. But we need a way for the computer to know where are these particles so that we can use them to make our 3D map, right? The kind of naive first approach that generally you'll take on basically all of your data sets is blob picking. And the way we blob pick is we just go and look at every single part of the micrograph and we say, how much does this look like a blob? And if that sounds kind of dumb, it is. It is a very sort of sledgehammer way of picking particles. You are literally comparing a Gaussian blob, like you see on the bottom there, to every single position in the micrograph, like you see at the top. Everything that correlates a certain amount with that Gaussian blob, it's called a particle pick. You say, that's a particle. Next pixel, no. Next pixel, no. Next pixel, that's a particle, right? So you end up with a ton of picks, right? You're just kind of, usually you'll end up tiling the micrograph, basically. And so you need to, well, let's back up. So now that you have those picks, you're going to extract them, okay? Extracting is how we actually get to a usable image of a single particle. You're going to get particle extraction as taking a square cookie cutter and just cutting out the little square of the micrograph that surrounds the place you said was a pick, okay? So now we'll have a separate image of every single particle. Why do we do this, right? Why don't we just use the micrograph as is? There's two big reasons. One is that doing all the calculations, rotations, translations, calculating these correlations is just going to be easier with a very small square image than trying to use these 4,000 pixel wide micrographs that we have. The other reason is when we extract particles, we can change the pixel size, right? You can imagine in Photoshop, like dragging the diagonal slider on your image, right? We can make the image bigger or smaller. Well, only smaller, really. And the reason we would do that is to speed up calculations. Okay, so this brings us to the next kind of important decision point when you're processing your data, which is how big should my box be? And we got some questions about that with CTF already. There's some other considerations too. So to answer these questions, let's all look at a picture of my cat. You can ask me a question later about her, but the important thing is we've extracted a square image of my cat from a bigger image I took of her laying on my yellow comforter. Okay? This is what I'm going to call our base extraction. Nobody count, but I think there's 128 pixels on each side. People you'll see will generally use powers of 2. There's a bigger list from the EMAN website that you can pull up to find the good box sizes. But let's just say 128 pixels for now. If I wanted to, I could extract with a smaller box size, so a smaller final image, but the same physical size on the micrograph. Okay? This is called downsampling. Downsampling is when you have the same physical area represented with fewer pixels. Okay? The reason you might want to do this is it'll be faster to perform your calculations, right? Now, instead of 128 squared pixels to consider, I only have 64 squared pixels to consider. So that's 4 times fewer numbers to think about, which means it'll be faster, right? We love fast. Why wouldn't you want to do this? There's something called the Nyquist limit, which says the finest resolution, the highest resolution that you can represent in an image is twice the pixel size. So if your pixel size to start with is 1 angstrom, your final 3D map will never, mathematically, will never have a resolution better than 2 angstroms. Okay? So if you downsample by 2, now your pixels represent 2 angstroms in physical space, which means your map will never have a resolution better than 4 angstroms. Okay? So the speed costs you something. You can actually see a representation of this, nice choice, because if you look at her ear, she has this dark band of fur right on her ear. And you can see that in the base extraction, but it's gone when we've downsampled because that band of fur is 1 pixel wide in the base image. So when we double the size of the pixel, we've lost the ability to resolve that little band of fur on her ear. Okay? I think that little band of fur is cute, and so I want to be able to see it. But the nice thing is I can extract my images at the downsampled size, do all of my particle cleanup, look and make sure I've only got good images of my cat, and then go back and extract at the full size so that we have the good pixel that lets us see that high resolution feature on her cute little ear, right? So this is the kind of big thing that I want you to take home from this slide is I think it's tempting to say, well, I always want to get a high resolution map at the end, so I always want to extract a full size pixel. I would recommend you don't do that. I would recommend you always start by extracting with a pretty downsampled box so that things are fast in those early stages where you kind of don't care what the final map looks like. You're just trying to find the good particles. And we'll talk about this more in the case studies. And then once you've found the good particles, you can re-extract, right? You can go back through with your cookie cutter, but only cut out the good positions and do those at full size, okay? The other thing to think about is how big is the actual extraction box? So here on this third image, we've taken a bigger physical area, right? You can see more of the comforter. You can see that little fold in the bottom left. But the pixel size, greater physical size, which means the box is bigger because we've kept the pixel size the same, right? Each pixel here still represents an angstrom, but now we're looking at 256 angstroms, so the box has to be bigger, okay? So these are the 2 levers you have to play with when you're extracting an image. How big is the box in physical space? And that you're thinking about things like, does my particle fit in there? A, and B, does the CTF fit in there? And then there's the question of how big is the box kind of in image space? And that's things like, do I have room in my hard drive? How fast do I want this to be? What's my final quality of my map going to be? Okay. Like I said, generally people do twice as big as the particle for the box size. And then how big you downsample to will depend on what you're doing. Early stages, I often downsample to 128 pixels, kind of no matter what I'm doing. And you can go even further than that, although you may not want to. And then when you're coming back through and re-extracting, you can either do full size, or if your particle is very big, you can pick a pixel size so that twice that pixel size is kind of a resolution you expect your map to hit. So maybe you'd extract at 1.5 angstrom pixel size if you wanted to get a 3 angstrom map. So now we've got images of our particles, but remember, when we picked these particles, we were really dumb about it. We were just saying, how much does it look like a circle? So we're gonna need to come back through and do some classification, right? We're gonna say, is this particle actually good or not? But the particle images themselves are so bad that we need a way to kind of look at all the particles at once and find groups of particles that are good and groups of particles that are bad, okay? We're gonna use 2D classification to do that. So I'm gonna introduce some terms here. A class is a group of particles which are more similar to each other than they are to other particles. And the important thing to remember about a class is that it is entirely something that the software has found. We have no way of knowing ahead of time what a class is. And so the class will depend on how many classes you told it to find, how clean is your image stack to begin with, how many particles do you have per class you requested, right? This is where I'm going to really kind of try and define a pose. The pose is one of the most important concepts in cryo-EM. I'd say arguably the most important concept in image processing. So I really want to hit this home. A pose is where we compare a reference to a particle image, and we say what rotation and translation do we have to apply to that reference to get it to match the image. I'm sure you could find other definitions of pose, but that's the one I'm going to be working with here. A reference, that's something that we're saying, this is like what actually created this particle image. The reference is like the Platonic ideal of the particle. The pose is how do we rotate and translate that reference to make it match the image. So in 2D, our reference is going to be just an image. So it's easy enough to say how much do we rotate the image and how much do we translate it. In 3D, the reference will be a volume. So we'll rotate and translate that volume and then project it and compare that projection to the image, because you have to compare 2D and 2D, right? So pose is really important. And if you have any questions, once we get to the question part, I really encourage you to ask any questions you have about pose, because I'm going to be talking about it a lot, okay? So a class average is how we get our reference for 2D classification. So a class average is we take all the particles in the class, we line them up, right? We apply their poses and then squish them together and average them, okay? So by averaging them away, or by averaging them, excuse me, we get a much higher signal to noise ratio for that class than any individual image. Because ideally in the class, the particle is always the same, but the noise is random. So as you average it, right, the particle is the same every time, so averaging it doesn't change it, but the noise is positive sometimes, negative other times, big, small, whatever, so it goes away, okay? So like I said, I'm really gonna hit this home. A pose describes the rotations and translations that you have to apply to a reference to make it match the image, okay? Every particle image will have a different pose for a given reference, right? If I'm standing here, and I'm also standing like this, those two poses are different because they're different images, right? So here's 2D classification. And I'm gonna play this animation. What this is we're starting at iteration 0, and what we've done here is we've just averaged the particles together randomly. And the reason I'm sort of showing you this algorithm, even if you kind of know it already, is that this is also basically how the 3D one works. So we can just start here with 2 dimensions. We've basically just averaged together, we've put the particles in random classes and we've averaged them together with no pose, just random whatever, and we'll see what it looks like. You can see here, they're just blobs, right? And that makes sense. When we picked these particles, we know there's kind of something in these images or we wouldn't have extracted them. So there's kind of something and it's kind of in the middle of the image. And other than that, it's just noise. So we have kind of something and it's kind of in the middle of the image, but otherwise it's just noise. Then what we're gonna do is we're gonna take all of these references and line them up to each particle image. Okay, so each particle image will have its pose for all of these references, right? So we'll have all the particles times all the references, number of poses. Then we say, for every particle image, which class matches the best, right? So we've compared it to all 50 of these classes, but one of them, just by random chance, this noise is going to match that particle image best. We're going to say this particle belongs to that class. And then, once we've done that for every particle, we average them together. But now we've actually lined these particles up, right? We've given a pose to every particle. So when we average them now, we'll get something that looks a little bit more like the particle. And then we do it again. We say, all right, now with all our new images, right, align them. The pose gets a little bit better, which means our reference gets a little bit better, which means the pose gets a little bit better, okay? So we'll walk through the iterations, and you'll see pretty quickly how these classes change, okay? I've highlighted a few in green that are going to turn into good classes. I've highlighted a few in red that are going to turn into bad classes. What I want to emphasize here is that they look exactly the same because it's just noise. But as we get these poses, some of them will start to look like particles. And then once they look like particles, they'll get more and more particles in them, okay? Here we go. So you can see that we start with this random noise, but then through this process of iteratively lining up these particles and updating the reference and lining up the particles and updating the reference, they get better and better. I think this class average in the bottom left, I think, is especially fun because it starts to look kind of like the GPCR that makes these class averages, but then it starts to drift off center. And once it does, all the particles that used to go into that class say, I don't really look like this class anymore. I'm going to go over to this class, and boom, now there's no particles in this class, and it looks like garbage, right? So it's important to remember that during these iterations, the poses are changing and the class assignment is changing. And then because the poses in class assignment are changing, the reference is changing, which sort of causes this kind of feedback loop, right? Like I said, this was 2 dimensions. The process is basically the same in 3 dimensions. When we align an image to a reference, we find the pose, which has the least error between those 2 things, between the image and the reference or the projection of the reference. Typically, we iterate this process. So we do it over and over and over again. And then each iteration improves the reference, which means the pose gets better, et cetera. And then once the reference stops improving, this isn't always true. Some jobs do a certain number. 2D classification is one of them. But generally, once the reference stops improving, so once we say, you know, even though the poses are changing by half a degree here and there, the reference isn't really changing very much, I think we're done, right? That's what we call convergence, when the reference has stopped changing. Okay? So we're here to get rid of junk, right? 2D classification, we're going to get these class averages. And what this means is each of these class averages, right? We've found the pose, we've lined them up, we've averaged them. You can see how many particles is in each class, right? Here, we've got 24,000 particles that make up that class. But now this is recognizably something, okay? This class in the top right looks like our particle. So we'll say that's a good class, we'll keep it. That means all of the particles that are in that class, in this case, 34,000, those are good particles, we'll keep those. So we didn't have to inspect those individual images, right? But remember that these classes are made up of those bad individual images. Just by averaging them, we've got something we can actually assess, okay? This class is bad. It looks just like an empty micelle. So we'll discard this class, and we're sort of assuming that all the particles in that class also are just empty micelle particles. Okay. Here's the first one of these. So in a typical 2D class, the reference, right, the class average, is some particular viewing direction of the 3D particle, okay? What is the reference in a junk class? All right, in the one I just showed you, it was a micelle, but sometimes you'll get classes that are just kind of noise or something you can't really tell what it is. What is the reference in a junk class? Would the algorithm that I've just described to you treat that class any differently than a class containing a clearly good particle, right? Is there any difference between the two? And if not, if it doesn't treat them differently, what would cause a particle to end up in a junk class? Why does a particle get put into a junk class and not a good class? So you'll have a minute to just kind of think to yourself, and then I'll let you know when you have 4 minutes to talk to a neighbor. Okay, so why don't you go ahead and find a new friend and discuss junk classes. Okay, thanks everyone. That's awesome. I'm really excited to hear everyone talking to each other about cryo-EM. That's all I do on the weekend, so it's good to know I'm among friends here. For the sake of time, we're gonna have to move on, but during lunch you can talk about junk classes too. As I said, I'll kind of answer these questions, but remember, this is just sort of my phrasing of the problem here. But I think the key concept is that there's no such thing as a junk class sort of set in stone, right? We say they're junk classes because we look at them and we say, that's junk. But the algorithm doesn't know, a good particle from atom, right? It just says these images look like each other, okay? So junk particles end up in a junk class because they look like that specific junk class more than any other class average. And that's the important thing. you're always putting a particle in the bin that it looks the most like. And so you need to have a bin that looks like that junk, okay? Because of that, I and others in the cryo-EM community generally recommend doing a lot or most or all of your cleaning in 3 dimensions. And the reason for that is we are really, really bad at, in our head, taking a 3D volume, rotating it, translating it, and then projecting it into 2D space, okay? That's just not something we're good at. Well, we'll talk a second. Sorry, I forgot my own slideshow. Okay, so first, we want to clean in 3D. How do we do that? We have 2D images. How do we get to the 3rd dimension? The key concept here is called the forward model. Okay, so this is kind of a way that we model the creation of cryo-EM images that we record. Okay, so the forward model tells us, or says, that every image in the particle stack is a projection of the same rigid volume in some pose. I've already talked enough about poses, corrupted by the CTF and by noise, right? The forward model is how we say the images are formed. Now, I'm sure when you were reading this, you thought to yourself, well, every particle is a different volume, so they're not really the same thing. We know they're not rigid. Even rigid things are not rigid, right? This is a model. And a lot of the pathologies you see in your maps are because your particles are not conforming to this model. Maybe you have more than one volume in there. There's something bound to a drug or not. Maybe it's flexible, right? So it's not rigid, right? Maybe you don't have a very good pose. And so the work you're doing with this model is work. But this is how we model the creation of these images. And then based on this, we can use something which I won't get too much into, but it's called the central slice theorem, if you're interested, that says if we know the pose of a 2D image, we can basically use some clever math and the Fourier transform to put it in its proper place in 3D. And so as long as we have all the different orientations, we can get a 3D model from 2D images, as long as we know the poses, and as long as we assume this is how the images are made, okay? We'll talk a bit later about data sets that break this model, and we have to use different jobs that use a different model, but this is kind of the fundamental one, at least for CryoSPARC. The nice thing about this is that since we can get the CTF, we can model the CTF from micrographs, right? We don't need to know anything about individual particles to get the CTF, like Ollie was saying. And the noise is kind of right there. It's a little more complicated than that, but for all intents and purposes, we can model the noise directly from the micrographs as well. We can treat the volume as purely a function of the particle image pose, okay? So once you have the poses, you have your volume. It's really, really nice. And it's also why I've been really emphasizing thinking about poses. Okay? So here's how this kind of works in 3D. And again, like I said, it's very similar to how it works in 2D, okay? We start with a volume and our particle images. These are real particle images. It's the same image. On the right-hand side, I've low-pass filtered it very significantly. But this is really what your images look like, okay? So bad. So we start with the volume and our particle images, and we check basically all the different rotations and translations, and we find the one that when we project it, it matches that image best, right? Here's our reference. We find its pose that matches best. Then those images, we store that pose. We move on to the next one. Rotate, find the pose that matches best. That particle's pose gets stored. We move on and on and on and on until we've done this for all the particle images, okay? So every particle will have its own pose, right? We say the volume, that one rigid volume, was in this particular pose for this particular image, okay? Once we've done that for all of our images, we can now come back and use that central slice theorem and the forward model to say, now that we know the poses were this for all these images, what should our volume look like? And now we have a better volume, right? And it's the same thing as we did with 2D classification, where we iterate this until that volume stops improving, okay? So that's kind of the fundamentals, right? We collect raw data as movies rather than micrographs for the reasons we described. We collect it low dose. We're going to pick particles by just measuring how well they match some image, okay? By default, in the blob picker, we're just using a little blob. We then extract out those particles, make processing faster and easier, or to capture more of the CTF, whatever it is. To align a particle, we find the pose that makes it match its reference, okay? And the volume is the output, right? Always remember the volume is not the data. The volume is a product based on these assumptions, the assumption of the forward model, the assumption of your pose, right, et cetera. The volume is the product of your work, okay? I think here would be another place to take a break for questions before we finish up this first fundamentals section. Oh, sorry, someone wanted to take a picture. Go ahead.
Can I?
Hi.
Hi. Oh, so I'm talking about like CryoSPARC. You have talked about the blob peaking.
Yes.
So if I want to get the micrographs with some big oval shape or filamentous protein, is it still possible to pick the particle with the blob picker?
Yes, it is. So there's in the blob picker, there's different modes. The one I described, I think, is just called blob. And there's also elliptical mode, which sounds like kind of what you would want to do. And I actually, in the case study that we do next, I'll show the parameters that you can use to change the size and shape of that blob.
Okay.
So you can kind of fine tune it. And the blob picker, like I said, is kind of a sledgehammer. So ultimately, you'll probably want to go back through and use template picking, which we'll talk about. But that's where you use an actual image kind of your protein, more or less, to pick rather than these random little blobs. But there is a way to pick like a stretched out elliptical blob instead of a circle.
Okay, thank. I just have one follow-up question. So, like for filamentous protein, sometimes when we freeze the sample, we see like some particles or filaments are edge of the hole, so that we have to take the image with the carbon support also. Does it hamper the CTF estimation? Like, when we do motion correction and other things, does it hamper the total overall resolution?
Yeah, it's a good question. So like CTF and things actually, I would say generally will be okay with a carbon edge. In fact, carbon will give you a little bit more signal to find the CTF. Might be the wrong focus, but the real thing is alignment is going to be hurt by the carbon edge. Because if you think about what we're doing, right, we have our reference that just has your filament in it, but your image has this huge chunk of carbon.
Yes.
And so when you try to line up your reference that doesn't have that carbon to the image that does, you might end up putting the filament down on the carbon because there's just so much contrast there from the carbon. So you'll get more hurt in your alignments than the early like pre-processing stages.
Thank you so much.
Hi. Sorry.
Oh, thanks.
So when you're talking about the 3D volume refinement, you were basically saying how, the reference volume and then the images, the particles in the images are like their poses are aligned to match the reference volume. But, before that, to get the reference volume, you have to do like the ab initio reconstruction stuff, right?
Yeah.
Does that work under the same principles, or how do we actually generate that first volume from the 2D classes?
That's a great question. And I'm glad you asked it, because I sort of skipped a step where I said, yeah, you line up your particles to the 3D volume. It's like, where did that 3D volume come from? And yeah, ab initio is how you would generate that volume. In principle, it is similar. It's a different algorithm. It doesn't do this kind of iterative. I mean, Ollie should maybe talk about it, but it basically is kind of the way I would give you sort of an intuition for what it does is it is trying to get as fast as possible to something that believably looks a little bit like your volume. I'm sure Ollie will give more mathematical description, but basically it's going to give you a bad volume that looks enough like your particles that you can start to do this alignment process. And then maybe we could talk about the differences between the two algorithms. Yeah.
I can just add a tiny bit to that. I think that's a perfect intuitive explanation. Ab initio reconstruction, the method that it uses is a little bit different than this iterative concept of taking a reference, aligning the poses, and then recreating the reference, only in that it uses the concept of stochasticity, random selection, looking at only a few images at a time to quickly jump around and make changes to the volume so that it starts to look more and more like the data. And this is a well-established statistical technique called stochastic estimation or stochastic gradient descent. There's a whole set of varieties of these algorithms that use the concept of random selection to help optimize difficult problems like this Cryo-EM initial reconstruction problem. We plan not to get into the details of that here since there's a fair bit of content about ab initio reconstruction existing already. But intuitively, I think Rich's explanation is exactly right. It helps you, it allows you to take your particle images, not knowing their pose, not knowing the 3D reference, to construct an initial model. Ab initio reconstruction won't be able to resolve high resolution details or really any other interesting stuff except for some heterogeneity, which we'll talk about later as well. but it allows you to get initial 3D references that then you can plug into this iterative loop that let you get all the rest of the way to the end of the problem. But it works using a different statistical estimation technique than this iterative process.
Yeah, and there is our guide page on ab initio has some good information too. Go ahead. Yeah.
Okay. Hi. I was curious about how many classes, like when you decide like how many do you want to pick? Because if you have so many classes, you might be able to capture like different states, but like then you might not have enough particles to average. So how do you pick, like, especially if you have a super heterogeneous or flexible particle, what that range of error is, and is the number of classes correlated with resolution? I don't know if that makes sense.
Yeah, no. So it's a good question. How do you pick the classes, basically, given all these different problems? And I think your point is exactly right. That I think ideally you'd have every particle is different, right? So ideally you'd have 1 million classes, but then you lose that benefit of averaging them together. So you're always going to be trading off the kind of resolvability or resolution of your class, not like formal resolution, but like how much can I see what this is against how high quality is that signal to noise ratio. And I think by the end of this workshop, you're going to get tired of me saying it depends on the sample, but unfortunately it depends on the sample. Generally, I think these days, with the absolutely wild sizes of cryo-EM data sets, you will never have too many classes, frankly. I think you can really go crazy. Then the question is it worth your time? Because more classes also makes the job take longer. And as I said, and as I'll show you in one of the case studies, 2D classification is really these days just not the best tool we have for separating out heterogeneity. You mentioned flexibility. There's much better jobs for that use 3D. And so that both makes it easier on you to say, you know, if I'm waving my arm like this, it's easy enough for you to tell right now what's happening. But if I like look edge on at you and go like this, and you're looking at this black and white thing that's 2 pixels across on your monitor, right? And so in 3D, you open it up in Chimera X and you're like, oh, he's waving his arm. Great. So that kind of technique, I think, is much more powerful for the kind of thing you're describing. 2D classification, I really imagine like standing half a mile away from an animal and squinting and saying, is that a cow or a sheep? Not really very helpful. But if you're saying, is that a cow or a tree? That's the kind of thing that I think 2D classification is best for. So I think generally, I personally typically use 200 classes to start. That's just a random number. You can pick basically whatever you want. Depends on the GPU, depends on the particle number, et cetera. But I think these days, some classes is kind of the answer.
Okay, thank you.
Yeah, hi. So what I'm asking is that, so you talked about 2D class. So let's consider a situation that you have prepared your sample 100% pure, and you do not have about the idea of the structure of your sample. So that's why you are going to this cryo and all those things. So that time, how you will go like to select the 2D classes, because you have the idea about the particle diameter, but you do not the idea about its 3D orientations and all those things.
Yes.
So for that case, so is it a trial and error method to do the class 2D class, or anything else is there?
That's a really good question. So basically, if you don't know what your particle looks like, how do you know what a good class is? It's you know, it's a good question. I think my sort of recommendation, if you have no idea, and you know, I think these days you could, I'm going to say a dirty word, you could look at AlphaFold and kind of mostly know what it looks like, right?
That's
but assuming you don't want to do that, you don't like Google, I don't blame you. I think if you look at these 2D classes at the last iteration where they're done, right? You can still, even if you don't know what a GPCR with a fab looks like, you can say, that's bad, right? And you can say, that's good, right? And what I mean there is you're looking for good contrast between the light parts, like the parts that are a particle, and the parts that are just background. You're looking for there to be no like spikes that shoot off randomly. I don't see any here, but you know, if it's a lot of noise that's been overfit, you'll have these spikes that shoot off into the background that aren't a physical thing, right? And you know, these things that are just random speckly backgrounds. So I think if you don't know what your particle looks like, you still kind of know what not a particle looks like, I guess is my guidance.
So one more, just one more. So you mentioned about this size of the box and resolution, all those things. So this size of the box, because you are imposing in the CryoSPARC software, the size of the box based on your particle size. So is there like we have to give 2 times or 1.5 times or 2.5 times of the your particle size? Is that kind of any formula available, or people can use that for the making this box size with respect to your particles?
Are you saying like within CryoSPARC,
yeah.
does it calculate that for you, or?
Yeah. So basically you are giving 2 times of the particle size. Yeah, so
that would be my
it-
recommendation, but
Okay.
you're in control of how big the box is when you extract it. Is that, I'm not sure I'm answering your question.
Yeah, that one.
Yeah, okay. Great. I think
So
maybe let's do one more and then we'll do whatever's left at the end.
I have a question with the picking.
Yes.
So my protein is kind of like a dumbbell. Say one side is rigid, the other side is more flexible.
Okay.
Whenever I pick particles, it only picks the rigid part. It never picks the middle of my particle. Is there a setting in blob picking that I can use to shift it without having to go changing it further down the line, like shifting the center of the particle or something like that?
Yeah, so that's tricky. If you think about what you're trying to do from the micrograph's point of view, right? From a 2D class, it's easy enough to say, like, I want this to be shifted over here, like extract that part, right? From the micrograph, you would have to come in and rotate and translate the particles and extract them from that new center, which is possible. Generally, that's the kind of thing that I would recommend waiting until you're in 3D to do. And we have a job that's called volume alignment tools, I think would do it, where you pick basically a new center of your volume, and then you can re-extract the particles from that. The reason I recommend waiting until 3D is that your alignments, your poses in 3D are going to be just better generally than your 2D poses. Even if you have this flexible bit, you know where that flexible bit should be. So even if you can only see the rigid bit, if you see, you have your dumbbell and you have the weight on this side and the weight on that side, and the map disappears halfway along that connecting rod, you could still say re-extract particles in the middle of this rod. Even though you don't know what's on the other side, you know it's there. And then doing that in 3D, you have the better poses. You've already filtered out a lot of junk that doesn't even have the rigid part, right? So generally, I'd recommend using 3D. And then, yeah, volume alignment tools and re-extract. And I think there's stuff on the guide for that as well. Yeah, thanks. Okay, like I said, any more questions? We'll do them at the end for sure. All right, we were here. Oh, another one of these. We love these. Okay, so now another one of these little questions. I've described the refinement process to you so far, where we have a reference and we line up our images to that reference and update the reference, blah, blah, blah. What would happen if I got a reference, right? We had a good question, where did that reference come from? I have some 3D reference that I provided, and I have a particle stack that's pure noise. It is just absolutely blank white noise. What would happen if I told the algorithm to line up those white noise images to our 3D reference that I provided? I said, I went to AlphaFold and it told me the protein should look like this. Here's a stack of pure white noise. Find me a map. Right? Consider that the algorithm looks for the pose that has the highest correlation between the image and the reference. Okay? So again, you'll have a minute to just kind of think to yourself, and then you'll have 4 minutes to talk with your new best friend. Okay, so go ahead and talk about what would happen if you had this particle stack of pure noise and you tried to line it to a reference. All right. Thanks, everyone. Thanks for the lively conversation about our just random noise particle images. It's time for us to talk about validation. And if you look at the schedule, there's a whole thing about this later, so we're not going to go into too much detail here. But it's a really important concept, so I wanted to make sure I hit it during this first part on fundamentals. And the kind of take-home message here is that right now we don't have any way to control for overfitting, right? We're telling this algorithm, there isn't GPCR in this image, find it, okay? It's going to. In CryoEM, you'll often hear people refer to this as Einstein from noise. There's a famous paper that kind of coined this phrase, where you can find a picture of Einstein with his tongue sticking out in random noise. So I actually really did this for this workshop. Here's our template. We're going to find Einstein in noise. Here's the template, and we're going to use our particle images. This really is pure Gaussian white noise. I generated this by hand. No, I'm kidding. So here's my particle image. Here's the template. I'm going to align it. That doesn't look very good to me, but we have lined it up, right? And actually, I got kind of lucky. This really is random. You can see there's this faint little dark bit of the noise here where my mouse is, and it lines up a little bit with Waz's hair here, and that's what's happened, right? We've just random chance. We've lined up this bit of noise. That's one image. Here's 6. Okay, that's still noise. Here's 48. Still pretty noisy. Here's 337. Okay? This, I'm not joking. This is random noise that I generated and lined up. You can start to see the collar of Waz's shirt, maybe his glasses. Here's 2,000. Okay? That's pretty convincing. Here's 16,000. In a CryoEM data set, 16,000 images is not very many. You can see with random white noise, if we give a really high contrast, high quality template, you can find whatever you're looking for, okay, just by lining up these images. No image in this particle stack is something I downloaded from slack.edu or whatever. I just lined up random noise until I got what I was looking for, okay? It's important that we don't do this because we want to find out what's actually there, right? So how do we control for that? Right? When we're giving these references, how do we avoid just finding what we tell it to look for? What we can do is low pass filter the reference, right? If we don't give the high frequency parts to the alignment algorithm, any high frequency parts it finds are really there because it didn't have them to line up to. The question is, how much should we low pass filter the volume? You don't want to just kind of guess, I don't know, 10 angstroms feels right? You want to have some kind of method to find the right number. The answer is we do something called the Fourier shell correlation, okay? So when we're making these maps, we don't just say, all right, all these particles in all their poses, average them together to make one beautiful map. We actually split them into 2 halves, half A, half B, right? The Capulets and the Montagues, whatever you like. And never the twain shall meet. They are aligned completely independently, and they make maps completely independently. Okay, so we have half map A and half map B. Over here is their Fourier transform. Okay, in Fourier space, the further you are from the center, from the origin, the higher frequency we're talking about. Okay, so a shell, a spherical shell in Fourier space represents a certain resolution band, right? 3 angstroms is this little sphere. 5 angstroms is closer to the center. For each resolution shell, for each frequency shell, we compare the correlation in Fourier space of the 2 half maps. Okay. Hence the name Fourier shell correlation. You'll always hear people say FSC, usually pretty fast. When you think about the correlation at a certain frequency, this Fourier shell correlation, in these half sets, anything that's really there should be there in both. So they'll correlate. Anything that's just noise you've overfit, because the noise is different in each of the 2 half sets, it'll be different when you try to calculate the correlation. Okay? So basically, by measuring correlation, we're measuring reliability. How much is this in both of our independent half sets? So at low resolutions, we're basically just saying, is there a blob there, yes or no? So they correlate really well. When we go to middling resolutions, we've started to introduce some noise, right? There's a bit of overfitting happening, so the correlation is not perfect anymore. Each of these half maps has been overfit a little bit to match its own independent template, but it's never seen the other one, so the correlation's a little bit lower. And then finally, when we go out to that Nyquist limit, right, Now they don't correlate at all because we're just overfitting noise, right? We're just taking noise or making it match noise, but since the noise is different, the correlation is 0. Okay, this is called the gold standard Fourier shell correlation, or GSFSC, where you have these half sets, you make the half maps, and you compare them. Okay, people will often quote the GSFSC resolution, which is the resolution or frequency at which the two half maps correlate at 0.143. There's a reason for that number that's not really worth getting into. Just know that if someone tells you, oh yeah, it's a 3 angstrom map based on the GSFSC resolution, that means they split it apart, they made these half maps, and then when the line crossed 0.143, that was at the 3 angstrom mark. Okay? The GSFSC is just a number. It's one number to describe your entire map. Please, 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 please look at your map, because it's really easy to get a map that says, yeah, 2.5 angstroms, great, man. But that just means the half maps correlate well out to 2.5 angstroms. You might download the map and see, this is horrible. I can't even build a model into this, let alone 2.5 angstroms, right? On the other hand, you might run a job and see that the GSFSC is the same between the two, or maybe it even got worse. But maybe part of the protein that you really wanted to model, maybe the other half of your dumbbell, is suddenly there. And now it's there, they don't actually correlate as well, but that's fine because what you wanted to see is what's that other half doing, right? The GSFSC is not the be-all, end-all. It's just a useful tool to warn us about what things we can trust and what things we can't. Okay? So we make our final map. Now we have our GSFSC. We make our final map by taking those half maps, averaging them together so we get better signal to noise, and then filtering based on how much we can trust each resolution. So filtering based on that GSFSC curve. Okay? What if there's more than one thing in the particle stack, right? We did 2D classification earlier. Now we're doing 3D classification. The principle here is the same. Okay? We're going to change the forward model a little bit. All right? So originally, the forward model says every image in the particle stack is a projection of the same rigid volume in some pose corrupted by the CTF and other noise. We're just going to change that a little bit to say every image in the particle stack is a projection of one of several rigid volumes in a pose, blah, blah, blah. Okay, so the thing we've changed is we've said originally they were all from the same volume. Now we're saying these particles belong to one of A, B, C, D, E, F, G, whatever. But the process is the same. So for now we have 2 references. Just like in 2D classification, we had n references, right? We line up both of these volumes, and they'll probably have a different pose, and probably one will match better than the other. So by default, most jobs in CryoEM won't just say, all right, this particle's either in A or B. They'll say, oh, it mostly looks like A, but it looks a little bit like B. So it'll kind of split the signal between the two. That's something you can switch on and off. You can make it do hard classification if you want. We'll talk about that more in the case studies. But again, you just go through all the particles in the stack, right? And you measure, you find the pose for both of your volumes, and then you say that particle belongs to the class it matches better once it's in that class's pose. Okay, so now every particle will have 2 poses. They'll have a pose for A and a pose for B, or n poses really, right? But you're doing the same thing. You're going through, you're updating all the poses, and then finally you update the volume. It's the same idea, but just twice as much. Okay? So now we've got our 3D maps, we've reduced overfitting, and we can handle multiple different types of sample, right? But have we squeezed every last drop of information out of these particle images? Okay, there's one last concept that we haven't really talked about, and that's symmetry. We all love symmetry. We remember it from kindergarten. In 3D, symmetry is a little bit more complicated than in 2D, but the same principles apply. Here is one of the proteins we're going to be talking about. This is TRPV1. This has C4 symmetry. What that means is it has 4 positions where it ought to ideally look identical, right? It's got 4-fold symmetry around one axis. In reality, almost nothing is symmetric, right? There's going to be this slight deformation so that those rotations are not perfectly the same. Because when you say C4 symmetry, the computer says exactly the same, when really we're saying they're the same, right? So how do we handle this? In some cases, it might be sufficient to say they're exactly the same. We call that enforced symmetry, where you take your particle, you create kind of 4 copies of it, but all those copies are rotated by some amount, so they're in one of the other poses, but you only use one pose. So you align it once, right? And you use that pose for all 4 copies, quote unquote. Then there's something called symmetry expansion, okay? This is where you really do still make all 4 of those copies, But now you treat them as totally independent particles. Okay, so they can have their own poses. This is called symmetry expansion. This is going to be more useful if the particle has some kind of asymmetry in it, right? So forcing it to be symmetric doesn't work, but it's still kind of mostly symmetric. Then there's something called symmetry relaxation, which is where you don't ever create those 4 copies. What you do is you say, this particle has some kind of symmetry-like thing that is like, in this case, a C4 symmetry. You find one pose, and then you check the other 4, right? So you find one pose, and then you say, all right, this is what I think the pose is, but I'm going to double check the ones that are related by 90-degree turns in this case. Okay? I just wanted to highlight those concepts for you. Michael's going to talk a lot about them, I think I am as well in some of the case studies. But if you hear people talk about enforcing the symmetry, you're aligning it 1 time, but you're using it n times, where n is the symmetry order of the particle. If you do symmetry expansion, you're aligning it n completely independent times, but you're using each of those alignments only once. And then symmetry relaxation is just checking the poses that are related by symmetry, but only using the particle once. You don't get that n-fold increase in signal-to-noise ratio. And I forgot to say that. That's the big advantage of using symmetry, is that a single image, in this case, it was 4-fold symmetric, right? Because we could use that single image in 4 different poses, we basically got to use it 4 times. So you really increase your signal-to-noise ratio when you have a symmetric particle. Okay? That is the end of the fundamentals part. So we have 5 minutes before our break, and if there's any more questions, we can definitely talk about those, and then we'll have a quick break and move on to the first case study.
So about the symmetry expansion, if I already did the non-uniform refinement with the C4 enforced, can I still use that result to do the expansion? Do I need to go back to the C1 heterogeneous?
Yeah, that's a good question. So if you've already enforced symmetry, whether or not your particles are symmetric, your map will be, right? And so whether or not you have to go back and do work in C1 symmetry depends on how asymmetric your particles are. So generally, I would recommend, if you want to do something with asymmetry, there's in the symmetry, in the refinements, actually, Michael, where is the break symmetry?
Yeah, so there is so we have in CryoSPARC, we have a job called homogeneous reconstruction only, and that one will take particles that you've previously aligned through a refinement job, and it will just generate a 3D volume from those particles as a kind of convenience feature to help in this case where you already have a symmetric reference and you want to go back to a C1 structure. We have a little toggle that can break the symmetry in a random way, and that will just take the particles and randomly assign them to one of their symmetry-related poses and then generate a volume from that. So that's a kind of convenient, quick way to take your volume that you've already started from, that's already C4 symmetric in this case, and go back to a C1 symmetric volume. But in cases where you can start from C1 and get a good structure with like ab initio reconstruction or refinement, you might have more success, in my experience, in that case, because in that case, you're giving the algorithm the chance to not enforce symmetry, and so it won't get locked into this like symmetric local minimum. Yeah, I don't know if that answers.
Yeah, I think that's a really important point that I was going to make as well.
Yeah,
I think it's tempting if you have a map that's like almost there and it looks symmetric to impose symmetry, and then you're off to the races, right? My recommendation is to kind of wait as long as you can to impose symmetry, because if you think about the alignment steps, right, your C4 symmetric map, even if the particles are not C4 symmetric, if the map is identical in all 4 of the poses, no one of those will be better than any other. So it'll be harder to break that asymmetry. So that's what I was saying about, do you have to go back to C1? If you have a really big asymmetric feature, that first iteration, there's no pose than any other, so it'll go to some random pose. But then the accumulation of noise might pull all those symmetric features into one instead of the others. So sometimes you can get away with it. But I recommend just trying to stick with C1 as long as you can. And I actually have one of the case studies goes into kind of this exact problem of starting with an imposed symmetry map and going to an asymmetric map. So we'll be able to talk about it more then.
Hi, just circling back to template bias.
Yeah.
We're talking about it in reference to like final validation and whatnot, but what about like, template picking, using the 2D classes as a template to go back to pick particles, or like the reference-based 2D selection where you give it a volume and then it assigns your 2D classes based on that? Should we also be worried about template bias in cases like that?
Yeah, I mean, I think you should always be worried about
Yeah.
it. The question is how worried.
Yeah.
So by default, CryoSPARC really aggressively filters the templates that you use for template picking, I think to 20 or 30 angstroms, and it also filters the micrographs themselves. So I would not change those parameters really, because you don't want to include that high resolution information in your template picking, because that's where the bias problem comes in, right? If you give a 20 angstrom 20 angstrom filtered template, and it picks a bunch of stuff that looks like your 20 angstrom filtered template, but there's no real information there, it's probably not going to give you a 3 angstrom made up map, because it didn't, the problem we have is we're saying we're aligning each pixel exactly right of the noise. Unlike real signal, which has these features, and so it has this broad correlation, the noise only correlates with your reference in exactly that one position, right? If I had taken, in my example, if I had taken my whole noise stack and rotated them 1 degree, it would have completely fallen apart. It wouldn't look anything like the reference, right? Whereas real data, if you randomly rotate them by 1 degree, it'll blur it out, but it'll still look like it. So that's kind of the big safeguard that's built in, is just low pass filtering everything. And then of course the GSFSC as well, having the 2 half sets, that kind of stuff. So I think you should worry about it. You should think about it certainly, but I wouldn't be too scared to use template picking just based on bias.
Great, thank you.
Yeah.
Just one point to add on that. I think often where a lot of users draw the line is using templates that came from your data,
Yes.
generated from your data through blob picking or Topaz, Cryolo, any sort of particle picker, versus templates that you've pulled in from an external source. For example, let's say you had, let's say you looked at the EMDB and you found a similar protein to the one that you're solving, and you generated templates from that structure using the generate templates job in CryoSPARC. That can work in some cases, but it is a riskier workflow for sure, because you're pulling in templates from a structure that isn't ultimately from your data itself. So yeah, that's something to keep in mind as well.
Okay, so I think now we are scheduled, I went a little over, but I think now we're scheduled for a break, and then we can move on to the first case study. Thanks, everyone.